erythrocytes, or typically known as the red blood cells or RBCs. These make up about 95% of formed elements, and they have a flattened, here we have a flattened biconcave disc, and it has a huge surface area to volume ratio. And so this is where form meets function, or fits function, where this is very important later on we'll talk about exchange of the different gases. So red blood cells lack a nuclei and mitochondria and other organelles as well. However, their main function is to carry out oxygen and to get rid of carbon dioxide. So we have about 5 million per millimeter square of blood of red blood cells in our total blood volume, and each red blood cell contains about 280 million globulin molecules, and so that allows for one erythrocyte to carry 1 billion molecules of oxygen. So hemoglobin is going to account for 1/3 of the cell's volume, and it consists of the protein globulin. And so here we have a molecule of hemoglobin, and so here would be 4 different chains of globulins that we find. And again, there are 280 million molecules of these hemoglobin only on one of the erythrocytes. And so each of the globulins is going to have a heme group, which here we have in the blue. And so here's a representation of the heme in this picture. And so each of the heme molecules is going to have a molecule of iron. And so iron is going to be what actually binds oxygen to it. So hemoglobin, the function of that is to transport oxygen with iron being required for it. And then the globulin molecules are actually going to transport carbon dioxide. So let me write that down. So CO2 for that. And then for iron, here we're going to have oxygen. Okay. So in order for us to transport our and exchange carbon dioxide is going to diffuse onto the red blood cells, and it's going to combine with water so that it's not toxic for our body, which will quickly dissociate into hydrogen ions and bicarbonate. So here we have carbon dioxide is going to combine with water, and then that's going to form carbonic acid, and then so it dissociates into hydrogen ion and bicarbonate. And red blood cells have carbonic anhydrase, which will reverse this conversion back if we need it, and to expel it out of the body within our lungs. And so carbon dioxide, about 7% is going to be dissolved in plasma, but then about 70% is going to be transported as bicarbonate ion throughout the body. So next, let's look at the life cycle of a red blood cell. So about 1% of all red blood cells are going to be replaced daily, and so this equates to about 250 billion cells per day. And so red blood cells, or erythrocytes, are going to be formed in the red bone marrow. And so we find these in different areas, such as the ends of long bones. We find this within the pelvis and sternum, and then also within the vertebrae. So then it's going to circulate within the bloodstream for about 120 days on average, or a range of 100 to 120. And then once it's done, it's completed its 120-day life cycle, then it goes and is phagocytized in the liver, and then also is destructed within the spleen. Then we recycle some of the products, especially heme, and iron is going to be the big one. So heme then is going to have the iron be broken down and then into its into the globulins, where iron is going to be transported back into the blood by transferrin, and then it's going to be stored in the liver by ferritin, okay? And so 25% of our iron is going to be found always in the liver, so that we always have some type of store. Then we have the heme is going to be converted into biliverdin and then into bilirubin, which is going to be secreted into bile in the liver and is excreted. And so for any of you that have heard of the condition of jaundice, where that's an excess buildup of bilirubin, and so that means that the body hasn't been able to eliminate that bilirubin. And so typically this is found with newborns, and that just means that the liver is still breaking it down. But usually with a lot of light, and it helps with getting rid of that bilirubin. And then lastly, what we have is then the membrane is going to have different proteins and globulins that break it down, and then we reuse those proteins to make new erythrocytes, and then those are going to be shipped back into the bone marrow so that more erythrocytes can be formed. Now typically with iron, where we get this is through our diet, and so it's a very conservative process where we don't need to have a lot to replenish what is lost, and so we typically get our iron from meat, liver, egg yolks, shellfish, legumes, and cereal. And then we lose iron though in sweat, urine, feces, and cells on the skin. And then also a big one is menses with females. So other dietary considerations with the formation and production of red blood cells is going to be folic acid. And so folic acid is a vitamin that is needed for synthesis of thymine, which is a nucleotide involved in DNA formation and cell division. And we get this in leafy plants, yeast, and livers. And so people who have folic acid deficiencies have decreased red blood cells. And then another one is vitamin B12, which is a vitamin needed for action of folic acid. And this one, though, however, is only found in animal products. And so vegetarians need to obtain this through external vitamin supplements so that we can have the production of red blood cells. So let's look into red blood cell production and control. And so big thing here is that when we have low blood oxygen, this causes the kidneys and the liver to produce a protein, or a hormone, which is erythropoietin. So this is going to be EPO for short. And so this is going to stimulate then increased red blood cell production. And what we have is erythropoietin is going to be secreted into the bloodstream, and then again by the kidneys, by cells in the kidneys, and by cells in the liver. And though the big thing though is it is secreted at a rate to replace lost red blood cells. So typically this is in a negative feedback fashion that we have, where then that's going to signal the red bone marrow to increase the number of red blood cells, and then so that we can increase the oxygen carrying capacity, again in response to low blood oxygen levels. And so what this does is the red bone marrow has these progenitor cells, and these become mature cells in order for them to become erythrocytes. So some of the causes of low blood oxygen is insufficient pumping by the heart, could be due to lung disease, could be anemia, or also high altitude. And so again, we have an then increase in EPO, and then an increase in red blood cell formation and production, and then all in all, that makes an increase in oxygen carrying capacity. And so erythropoietin will stop being secreted once we have increased the amount of oxygen within the blood. So this process takes about 3 days in total to occur. So next, with red blood cells, let's look at the different antigens and blood typing. So antigens are going to be found on the surface of red blood cells. to help immune system recognize itself. And then these are due to lipids and glycoproteins that are found in the plasma membrane. And then antibodies will be secreted by the lymphocytes in response to foreign cells and are typically found within the plasma of blood. So here we're going to have type A blood. And so on type A, what we have is A antigens that are going to be on the surface on the red blood cells, and the antibodies that it has are going to be B, anti-B. Then type B blood is going to have B antigens, and then it will have anti-A antibodies. Type O blood has no antigens, so there's no such thing as a type O antigens, rather it lacks type A or type B. And then though it has both anti-A and anti-B antibodies. And then type AB blood has both A and B on its surface of the red blood cell, but then it does not have any antibodies found within the plasma. So then think about which one would be the universal recipient. Would it be someone that has all the antigens, or would it have no antibodies? And so big thing here, type AB has to be the one that has no antibodies. And then type O blood is going to be where it has no antigens, and so this is going to be termed as the universal donor. And so this is important when we talk about agglutination. And agglutination occurs when we have transfusion reactions, and so if a person receives the wrong blood type, then antibodies would cause the red blood cell to agglutinate, or another fancy word for clumping. And so, for example, if somebody was given type A, or someone who was type A blood was given type B blood, what would happen is that the antibodies would cross-react, because again, if someone type B blood has B antigens and then A anti-A antibodies, that means that the antibodies for A would attack the antigens on the red blood cells, and so this would cause a clumping reaction. And then same thing if we did type B blood was given type A blood. So if this was the recipient and this was the donor, then we would have clumping here, where the B antigens would be clumped with those anti-B antibodies. So another consideration for red blood cells are the Rh factor, or commonly known as antigen D. And so these are either going to be present or not on the red blood cells, as these are just antigens. So we find these just on the red blood cells. So Rh negative does not have the antigen, Rh positive does have it. And so the Rh negative will not have the antibodies unless they're exposed to an Rh positive, either through a blood transfusion or during pregnancy. And so this is a problem, or can be a problem with pregnancy if, for example, the mother is Rh negative, but then is exposed to a Rh positive fetus, where that could be due to the father who has donated the Rh positive gene to produce that antibody. And so if that is the case, though, that the Rh negative mother has an Rh positive fetus, then a condition known as Erythroblastosis Fetalis in future pregnancies is where these antibodies from the mother that's been produced can cross the placenta and then attack the fetal red blood cells. And so that would cause clumping or agglutination. And so that agglutination would bind like at small areas, such as like within the capillaries and in the kidneys, and would prevent blood from flowing to certain areas. Luckily, in the 20s, there have been lots of diagnostic procedures where RhoGAM is a drug, pharmaceutical drug, that can inactivate the antigen, and so both the mother and the fetus are healthy and can survive. Okay, leukocytes, these are going to be the next type of formed elements, and these are white blood cells or WBCs, and so these are very different, where they are the only blood component that has complete cells, yet they are less numerous than red blood cells, making up only 1% of the total blood volume. This can leave capillaries in a amoeboid fashion, and so whereas red blood cells are restricted to the capillaries and vessels themselves, leukocytes can squeeze out of capillaries and out of that epithelia of capillaries to go to different areas within the tissues spaces. So the two big functions of white blood cells are, one, to protect the body against invading microorganisms, and two, to remove dead cells and debris from tissues by phagocytosis. And so with that 1%, what we have is a range of only 5,000 to 9,000 cells per millimeter cubed of blood. So we have two different categories of leukocytes. The first one are going to be the granular leukocytes, and so these include the neutrophils, which are most prevalent, eosinophils, and then basophils, and they're termed granular because they have a grainy-like appearance to their cytoplasm. And then the other two are agranular, which are monocytes and lymphocytes, which do not have a grainy-like appearance to their cytoplasm. So if we look at the differences here, again, the most common here are going to be neutrophils. And so these are going to have different peroxidases, different enzymes, and antibiotic-like proteins. And so these are going to destroy our bacteria. So let's write that down. Destroys destroy, and this is very hard to write on the computer. Okay, destroy bacteria. And so example of accumulation of dead neutrophils too is going to be of pus that we find within the bodies. So this is where we have cellular debris and fluid of infections that have congregated in a certain area. Okay, so next let's go to basophils. These are going to have more of a lilac and blackish purple appearance to them when we look at their cytoplasm. So this one is going to induce histamine, okay? And so histamine is going to have anti-inflammatory properties that acts to vasodilate and attract other white blood cells that we have. And so another one too is that it has heparin, which is going to prevent formation of clots. So this can be a problem when we need to induce blood clotting, and so heparin or basophils will not be recruited when we have a blood clot. All right, so then of the last granulocytes are going to be the eosinophils, which make up only 1 to 4% of our white blood cells. And so these are going to look very different, where they have a red cytoplasm to them. And these are going to be involved in the allergy response and inflammation. So this is going to lead the body in counterattack, especially against worms that we are exposed to. So then the two agranular site are the lymphocyte and the monocyte. And so lymphocytes make up 25% or more of the white blood cells. And these are going to, again, have a lack of that granules to them. So there's two different types. We have B lymphocytes and then T lymphocytes, okay? So the B cells are going to be stimulated by bacteria or toxins, and then these also give rise to plasma cells, which produce our different antibodies, okay? B for antibody. And then T cells are going to protect against viruses and other intracellular microorganisms. and are going to attack cells that are infected. These are going to be produced within the thymus. Monocytes are going to be less common, where 4 to 8% of them are circulating. And so these are going to leave circulation and enter tissues and then act as macrophages, or a little Pac-Man of our blood system. And so they're very mobile throughout the immune response. All right, and then the last of the formed elements are going to be the platelets or thrombocytes. And so these are going to be small fragments of larger cells of megakaryocytes, which are cytoplasmic portions of large bone marrow cells. These two, just like erythrocytes, lack a nuclei, and then they're very short-lived, though. They only last 5 to 9 days in circulation within their blood. So their function is to help with blood clotting in 2 different mechanisms. So the first is that they form a platelet plug, which seals holes in small vessels, and then also helps to format ions of clots, which helps to seal off larger wounds in vessels. They release serotonin, and then the big thing is that it helps with vasoconstriction. And so we have a lot of these where we have 130,000 to 400,000 platelets per millimeter cubed of blood. And so the process that is involved with the platelets and our red blood vessels for blood clots is when we have a tear that is formed within the blood vessel, the platelet starts to stick to the wound edges and to each other at the tear site. And so this is very important for homeostasis because it stops bleeding when there is a blood vessel is damaged, and so the damage then will induce also collagen fibers in the blood production. And so 2 other things that are responsible for, again, are serotonin. So serotonin is going to induce vasoconstriction, and then that will form this platelet plug that we have right here. And the platelet plug will have different platelets, it will have collagen, and then fibrin to bind together. But also, big one that we don't see in this picture, but ADP, adenosine diphosphate, is going to be found here, as well as von Willebrand factor, which helps to hold and make it activated and to make them sticky so that they clot and stay there. So fibrin is going to be the protein that creates this web-like fashion. So here we have our fibrin proteins that are insoluble, and that's going to hold the clot together until the vessel is able to heal itself. So one other vitamin too important for this is vitamin K to hold the fibrin together, and so vitamin K is very important for all these other different factors and growth factors to help hold that fibrin in place.